This is Ravindra Babu. Welcome back to my channel Silver 90s Bionet. In this video, we are going to discuss the second experiment, that is biochemical characterization of Griffith's transforming principle, under the heading of search for genetic material in the chapter molecular basis of inheritance. In the last video, we were talking about the Griffith's transformation experiment. The experiment concluded that the transforming principle or the genetic material is involving in the transfer of genetic characters from one bacterium to another bacterium through the medium in which the bacteria are growing. But Griffith could not able to define the biochemical nature of the transforming principle, but he thought that the transforming principle might be a protein molecule. So, in order to determine the biochemical characterization of the transforming principle of Griffith's experiment, there were three scientists called Colin, MacLeod and Oswald Avery, and third person is MacLeod and McCartey. These were the three scientists who conducted the in vitro experiments on the same organism, Streptococcus pneumonia, during the period of 1933 to 1944. By these experiments, they want to confirm that DNA is a transforming principle. Their experiments are completely in vitro experiments because they did not use any mice to identify the characteristic features, symptoms of the disease pneumonia caused by Streptococcus pneumonia. Here they just use the culture medium and grown the bacteria on the culture medium. Just by observing the type of colonies on the culture medium, they drawn the results. And if you look at the experimental procedure, the first four initial steps of MacLeod, Avery and McCartey were almost resembles the Griffith experimental steps, but the only difference is Griffith used mice to identify the symptoms caused by the pneumonia disease, but here they did not use any mice, just they drawn the results by observing the type of colonies grown on the medium. In the first step of their experiment, first they took R2 bacteria. What they took? They took R2 strains of Streptococcus pneumonia and allowed to grow on a medium. On this medium, they could find that rough colonies. What they found? Rough colonies. By this step, they came to a conclusion that R2 strains forms rough colonies on the medium. And in the second step of their experiment, they took S3 strains of Streptococcus pneumonia and allowed to grow on a medium. On this medium, they found that smooth and shiny colonies. They found smooth and shiny colonies. By this step, they came to know that this S3 strains forms smooth and shiny colonies on the medium. In the third step, they took heat killed S3 strains. To this heat killed S3 strains, they allowed to grow on a medium here, but they could find no colonies on the medium, no colonies at all, because though the S3 strains forms smooth and shiny colonies, but in this situation they were heat killed, so that they could not form any colonies on the medium. In the fourth step of their experiment, MacLeod, Avery and McCartey took the combination of live R2 strains and heat killed S3 strains, like that of the Griffith has done in his experiment. To this combination of both strains, they allowed to grow on a medium. Likewise, Griffith, they also expected that the formation of rough colonies on the medium as R2 strains are in live condition, whereas S3 strains are in heat killed condition, but it was not happened. Instead of that, they also got smooth and shiny colonies. Smooth and shiny colonies of S3 strains on the medium. Based on this, they came to a conclusion again that a transforming principle that is present in the heat killed S3 strains is being transferred to live R2 strains, which is making the live R2 strains to become converted into live S3 strains, so that they could form more smooth and shiny colonies of S3 strains on the medium. In order to know the biochemical nature of this transforming principle, MacLeod, Avery and McCartey, they wanted to continue their experiment further. But one more point, they came to know that the DNA is a heat resistant material than the other materials present in the living organism. So they thought that the DNA is might acting as a transforming principle, and this might be the reason behind the formation of more smooth and shiny colonies on the medium. Thus, in order to confirm this, they went for the fifth step of their experiment. In the fifth step of their experiment, they isolated the DNA from S3 strains, and to this DNA they allowed to grow on a medium. Actually, they were expected the formation of S3 colonies, smooth and shiny colonies on the medium, but it was not happened because the DNA itself cannot utilize the medium and cannot make a new organism. To utilize the medium, it requires a body like soul and a body. Here the DNA acts like a soul. Whenever it is having a body, it can enter into the body and can do anything. Whenever there is no body, the DNA itself cannot do anything. That is what happens here. This fifth step of their experiment concludes that the transformation requires not only a transforming principle but also a organism. And in the sixth step of their experiment, MacLeod, Avery and McCartey wanted to reconfirm that transformation process is taking place. For that, they took live R2 strains and heat killed S3 strains. Heat killed S3 strains. To this mixture, they are allowed to grow on a medium, and this medium is having anti-R2. Anti-R2 means this is the substance in the medium which inhibits or which kills the R2 strains whichever present in the medium. So, in the presence of this anti-R2, most of the R2 strains of the bacteria will be washed out from the medium, but there may be a few leftover R2 strains. Here, into this few leftover R2 strains, the transforming principle from heat killed S3 strains get entered. The transforming principle, it makes the R2 strains to become live S3 strains like this. These are the S3 strains. So, the number of S3 colonies or smooth and shiny colonies on the medium represents the number of leftover R2 strains. This indicates that the transformation is taking place. So, the fourth step says that the transformation is taking place. This is a confirmation. And fifth step says that transformation requires not only a transforming principle but also the organisms. And sixth step reconfirms that the transformation is taking place. And in the seventh step of their experiment, they wanted to determine the biochemical nature of this transforming principle. For that, he took live R2 strains and isolated DNA of S3 strains and to this mixture they are allowed to grow on a medium having anti-R2. So in the presence of this anti-R2, almost all the R2 strains will be washed out from the medium, so that this isolated DNA of S3 strain, it enters into that few leftover R2 strains whichever got the transforming principle, get transformed into S3 strains. The number of S3 strains formed on this medium is equal to this few leftover R2 strains. So here also they got few S3 colonies. By this seventh step of their experiment, they came to determine the transforming principle of Griffith experiment was a DNA molecule. By the seventh step of their experiment, we may think that the experiment of biochemical characterization of Griffith transforming principle is over, but it is not yet completed. Here, the three scientists of this experiment, they themselves got some doubt on this seventh step of their experiment. The doubts were: the sample of DNA isolated from S3 strains during this seventh step, was it having a purely DNA? That is the first doubt. And the second doubt was: the sample of DNA isolated from S3 strains was having any traces of RNA and proteins along with the DNA molecule. And the third doubt is, if the sample isolated from this S3 bacteria, if it is having traces of RNA and proteins along with the DNA, how can we say that the DNA is the transforming principle? Because there are the equal chances to RNA and proteins to act as a transforming principle if that sample is having these traces of RNA and proteins along with the DNA. These are the three doubts they got on their own experiments. So to make it clear, they continued their experiment further. So in the eighth step of their experiment, they took a live R2 strains and a sample of DNA isolated from S3 strains, DNA of S3 strains. To this mixture, they are allowed to grow on a medium. Okay, and this medium is having RNases. What the medium is having? RNases. Here the RNases are the enzymes which digest the RNA molecules if present in this isolated DNA sample of S3 strains. So that if the RNA molecule is acting as a transforming principle, they should not get any S3 colonies. But here they got smooth and shiny colonies of S3 strains. This indicates that the RNA molecule is not the transforming principle. In the ninth step of their experiment, they took same live R2 strains and DNA, isolated DNA of S3 strains and allowed to grow on a medium. And here the medium is having proteases. Proteases are the enzymes which digest the protein molecules. If any protein molecule, if any traces of protein molecules present in this isolated DNA sample, if the protein is acting as a transforming principle, here they should not get any S3 colonies. But here they got smooth and shiny colonies of S3 strains, S3 colonies. This indicates that protein is also not acting as a transforming principle. And in the last final step of their experiment, here the same, they use live R2 strains and isolated DNA of S3 strains and allowed to grow on a medium. Now to this medium they added DNases. What enzymes they added? DNases. DNases are the enzymes which digest the DNA molecule. Here the DNA molecule is actually acting as a transforming principle, so that no S3 colonies, This indicates that the DNA is acting as a transforming principle, because in the presence of DNases the transforming principle was digested, so that in the absence of the transforming principle the R2 strains remain as such R2 strains. They did not convert it into S3 strains, so that there is no S3 colonies. Thanks for watching this video. Please like, share and subscribe my channel.